In this lesson, we're going to talk about another one of the large organic molecules that's also a polymer, and those are the proteins. Let's start with why proteins are important to a living system. There are many different types of proteins that occur in the living world, and the 6 listed here are just a couple of the functions that proteins could have, some of which we're going to study in more detail in this course, including structural support, storage, transport, cell-to-cell communications, movement, and defense against foreign substances. Proteins are instrumental in almost everything an organism does because of these necessary roles that they play. And by the way, proteins come from the, the genetic code. So the code of life from the DNA is transcribed into messenger RNA in a eukaryotic cell, which then goes to the ribosomes, which then translate the messenger RNA into polypeptides, which ultimately might become proteins. One example of a protein is the group of proteins known as enzymes. And we will study this again in more detail later in the semester, but I just want to make note of this example because it's a really important group of proteins. These enzymes, they serve as biological catalysts that speed up chemical reactions in living things. And sometimes reactions won't happen without the aid of this enzyme, or at least they wouldn't happen in a timely manner that would be effective or useful for the cell. So here's an example showing you how the enzyme that facilitates the excuse me, the breakdown of sucrose through hydrolysis. Notice water is coming in and breaks down sucrose into its component monosaccharides, glucose and fructose. So in the processes that we had talked about previously of dehydration synthesis and hydrolysis, enzymes are sometimes required as shown here. So just kind of backstepping a bit, I had mentioned that the DNA code is used to first transcribe into messenger RNA, which then is translated into a sequence of amino acids, which is also known as a polypeptide. Polypeptides are by definition the polymers of amino acids. So the difference here is that proteins are often made of one or more polypeptides. So not all polypeptides are proteins. So process that for a second. Not all polypeptides are proteins because sometimes a single polypeptide alone is not enough to make a protein. That single polypeptide might have to be folded and perhaps even interact with another polypeptide to make a true functioning protein. So beyond the step of where the simple polymer is made at the ribosome of a cell, there's additional processing that sometimes has to happen before a functional protein is made. And this brings us to how we can characterize protein structure into these 4 different levels of hierarchy. The simplest form of hierarchy is the primary structure. This is just the simple sequence of amino acids. It's very linear. We're not talking about any foldings yet. It's just that linear chain of amino acids straight off the ribosome. And let me skip ahead and I'll kind of go back and forth here. So in this pictorial of that idea, that would be reflected here. This would be the primary structure. You can see if you zoom in to all those little purple circles, they have abbreviations for the amino acids. First one being glycine, GLY. Ending looks like an alanine. Okay, so that would be the primary structure. But most of the time, it doesn't end there for a protein. There needs to be some additional processing done. So let's go back and take a look at what secondary structure means. Most proteins have secondary structure, which includes coils and folds in the polypeptide chain, such as alpha helices and beta pleated sheets. So let's take a look at that pictorial again. At least we can see here what an alpha helix is, what a beta pleated sheet looks like. Kind of out of context here, but if you zoom in to this is a ribosome, which is partly made of protein, also RNA. You can see like this helix here, so that would be an alpha helix. And there's beta pleated sheets. So really what's happening is you have some interactions among some of these amino acids with other amino acids through hydrogen bonding, shown by the red dots there. That's what's holding those alpha helices and beta pleated sheets together. So we have the addition of hydrogen bonding at the secondary level. So the hydrogen bonds themselves, as well as what type of foldings result from those hydrogen bonding interactions. But again, we're just talking about within a single polypeptide. So now let's take it to the next level, the tertiary level, and see what changes here. Now everything builds, by the way. So at the tertiary level will be primary structure, the sequence of amino acids, and secondary structure, the coils and folds, in addition to interactions among various side chains of the amino acids, or R groups. The R groups are include functional groups like hydroxyl, sulfur, and so they can interact in ways beyond the hydrogen bonds, like ionic bonds, to add additional dimensionality to the polypeptide structure. So any additional interactions that we might be able to see here with the ribosome could be the result of tertiary structure. We don't really see a zoom in of what's actually happening there with the functional groups, the R groups, but we can infer that the ribosome has those as well. Okay, and lastly, the most complex and highest level of structure for a polypeptide, or protein rather, which includes multiple polypeptides, is the quaternary structure. So that is the definition actually. This is when you have more than one polypeptide that's interacting through those primary, secondary, and tertiary structures, in addition to the fact you have more than one polypeptide. Most proteins have quaternary structure. In fact, hemoglobin is an example of a protein with quaternary structure. Before I get to that next slide, I just want to take a moment to emphasize the importance of protein structure for function of the protein. Remember, there is a correlation in biology between structure and function, and we see that here with the proteins. And where it's most notable is typically when there's an error, like a mutation in the DNA, that led to the protein being made in a different way that changes its shape. And so no longer can it do its regular function. One example where this has been determined is with the sickle cell disease. We've used this as an example for other things, so I like to come back to this whenever I can. A very slight change in the primary structure of the sickle cell. Sorry, let me back up a second. What it means to be sickle cell is it means that the red blood cells are misshapen. That was actually caused, they found, by one single mutation in the DNA code. That's what led to the red blood cells being sickle-shaped instead of disc-shaped. So that single amino acid substitution changed the protein that resulted from that, causing it to have that difference in shape. Now, it is still a red blood cell and it can do some of the things that a red blood cell can do, but think about what would happen to the sickled cells as they're trying to pass through the human body. Sometimes through very, very narrow passages, like a capillary. They have the tendency to get clogged, and they can also cause problems in parts of the body where the blood is being filtered, like in the spleen. So people that have sickle cell have many symptoms simply because their red blood cells are not the normal shape that they're supposed to be. And again, it's all related to first the DNA having a mutation and that ultimately affecting the shape of the protein. So mutations can cause changes in the shape of the protein, but there are other factors as well. Here's a few other examples of factors that can affect the protein and perhaps even cause it to unravel or denature. For the environmental, we're looking at changes in pH, salt concentration or salinity, temperature, just to name a few things that are more common to discuss in this class. Once a protein is denatured, it's rendered inactive. It can no longer do its biological function. This is an illustration to show you what a normal protein might look like before and after it's denatured. So we will do some experiments in lab to actually demonstrate this and see whether or not a denatured protein can still be used to perform a particular function, specifically with enzymes. We'll look at that. All right, well, that's it for the lesson on proteins.